Okay, so I have some really good news, which is that I've set an alarm for 10 minutes before I'm supposed to end, but this time I've also set a second alarm so that when I'm supposed to end, I've got to end on time today. So this is my last lecture. I know you guys are staying here for another week, but I'm not. So I want to say thank you to all of you guys. It's been really fun to do this, but I especially want to say thank you to Deepa and Kavita, and I think probably we all want to say thank you to them, so please join me in thanking them. Okay, so we've been talking about this idea of sexually antagonistic selection. We got to the point last time where we were thinking about these polymorphisms existing on autosomal loci, and I had showed you a figure like this, or maybe I'll focus on this version of it, where if you have equal dominance, the range of selection coefficients where you can maintain the polymorphism is much narrower than if you have this so-called dominance reversal, where the allele that is bad for females is recessive in females, but the allele that's bad in males, the other allele, is recessive in males. And especially when you look at smaller selection coefficients, the parameter space where you can permit a protected polymorphism is much greater when you have this dominance reversal compared to the very narrow range without a dominance reversal. So here is perhaps the best example, maybe the only one I can think of off the top of my head anyway, from an Atlantic salmon. So the salmon reproduce in rivers, the eggs hatch there, and the offspring, they do their first part of the development in the freshwater, and then they go out into the oceans, and they grow and mature out in the ocean. And for females, it's better for them to come back at a larger size. So selection favors them to take 2 or 3 years to stay out in the ocean before they come back for their to reproduce. For males, it's better for them to reproduce at a smaller size and earlier. So for them, it's better to return after only 1 or 2 years out in the ocean. So it turns out that there are 2 alleles at a locus that affects the length of time you stay out maturing in the ocean. A so-called early allele, which causes you to come back early, and a so-called late allele, which does the opposite. And so we would expect that one allele would be favored for males and the other allele would be favored for females. And the really cool thing is they found this dominance reversal, where the early allele is dominant in one sex, and the other allele is dominant in. So here's where the early allele is dominant in males. You can see the heterozygote is like the early allele homozygote, and in females you get the opposite pattern where the heterozygote is like the late allele homozygote. So that's an example of dominance reversal. One might ask, how can you generally get this kind of dominance reversal? Is this something likely to be general? And the answer really is we don't know. But here's one cartoon of how it might not be so implausible, even though when we talked about it at first, it seemed like it would be implausible. So imagine you had some phenotype. This could be any phenotype. You can think of this as body size or whatever. And here is how that phenotype relates to fitness in one sex. And here's how that phenotype relates to fitness in the other sex. So then if you imagine that there's some gene, say this A locus, that additively affects that phenotype, so replacing a big A with a little a moves increases your phenotype by this much, and doing that again increases your fitness by, or sorry, increases your phenotype by the same amount. And then you ask, well, how does that, so the effect of the allele on the phenotype is completely additive. But let's ask how the allele affects fitness. And you can see if you were to focus on one sex, in this first sex, you would see that the heterozygote has a fitness close to the big A homozygote. And in the other sex, you would find that the heterozygote has a fitness similar to the little a homozygote. So it's simply a function of the curvature of the fitness landscapes acting on the additive phenotype underneath. So I was interested in this idea and was thinking about it in the context of the maintenance of genetic variation in the context of spatial heterogeneity. So this figure was made for thinking about two different populations, but exactly the same idea. Instead of calling that population 1 and population 2, you can call that males and that females. It's exactly the same idea. And I should say that these models or this model we've talked about for the maintenance of genetic variation with antagonistic selection between the sexes is extremely similar to analyzing models for the maintenance of genetic variation with spatial heterogeneity in selection, in particular the soft selection version. Okay. So this is, I wanted to show you, so I showed you this one result here that came from salmon for one particular gene. I've showed you an idea for why dominance reversals might be more general. And this is, to my mind, the best data of looking an attempt to look more broadly for effects across the genome of whether there's evidence of this dominance reversal. And this is the work of Carl Grisham and Jorn Arnqvist, the paper that came out earlier this year. And so they were studying this Callosobruchus maculatus, this seed beetle. And this is what's known as a diallel cross, where they start with these 16 inbred lines. So essentially, they've collected beetles from nature and then highly inbred them. So they have these 16 highly inbred lines. And then what they're going to do is they're going to cross males from those lines with females from all those lines, filling out all these cells. So all the points along the diagonal will be those inbred genotypes recreated. But anything off the diagonal represents an outbred genotype. Whoops. Right, okay, so you can do that, and then you can look at you can do the kind of classic quantitative genetic decomposition of the form that Guy talked about in his first lecture. Okay. Because there's different levels of relatedness, say along the rows and along the columns, and so you can make these statistical inferences about the underlying genetic components. So in this case here, so what I didn't say what they measured. What they actually, the phenotype they were measuring was male fitness and female fitness from the offspring resulting from those crosses. So here was their estimate of the additive genetic variance for fitness, and you can see the point estimate was quite low, is basically 0, and the confidence intervals certainly spanned 0. Not sure exactly how one should interpret a negative estimate there. But perhaps more interestingly, there's a large dominance component of variance, and also this quite large sex by dominance interaction component, which might signal. This is kind of an indirect form of suggestion of maybe that dominance works differently in one sex than another, right? So that's maybe a hint towards this dominance reversal thing being common. To get at that more precisely, these guys came up with something I had never heard of before, and which I thought was quite clever. And I just want to explain the basic idea of. They looked at to really look for better evidence of this dominance reversals in this framework. They did something called looking at array covariances. And here's the idea. So remember, all these diagonal points represent the inbred genotypes. And so the different color and size squares, just we can imagine those represent the phenotypes, right? So this one has a small orange phenotype, this one has a large green phenotype, and so on and so forth. So let's imagine line 1 as kind of our focal line that we want to look at. Now, let's imagine that strain 1 has a bunch of recessive alleles. If it has a bunch of recessive alleles, then when line 1 is crossed with these other lines, then the resulting phenotypes should tend to look like what the other line was on its own, right? So if I cross line 1, line 1 itself is this medium-sized white square, and line 3 is on its own is this big fat green square. While when I cross the two together, if line 1 has a lot of recessive alleles, then the offspring between line 1 and 3 will tend to look like parent 3. Okay, so that's essentially the idea that the diagonal should be a good predictor of the rows and the columns if line 1 has a bunch of recessive alleles. And if on the other hand, line 1 contained a lot of dominant alleles, then the diagonal will not be a good predictor of what's on the rows and the columns. So essentially, you're looking at the covariance between what's on the diagonal and the average of the row and the column. Okay, so that's why it's called an array covariance. So they did that, and remember they measured remember what's going on here. They're measuring 2 phenotypes. So far I've just been talking about, in this cartoon, as one phenotype. So they did this kind of array covariance for male fitness, and then they did these array covariances for female fitness. And you're getting a value for each line, right? So so in this example, I would see there's a very low covariance between the values here and the values on the rows and columns. So the array covariance for line 1 in this case would be something low, whereas the array covariance in the other case would be something larger and positive. So they did that. So for every line, they got its array covariance value for male fitness and also its array covariance for female fitness. So you would expect then, if there were no dominance reversals, if dominance was similar across the sex, between the sexes, then if a line had a positive array covariance for male fitness, it would also have a positive array covariance for female fitness. So now what we're going to look at is the correlation between these array covariances, the array covariance for male fitness and the array covariance for female fitness. And this is what they found. So here are the array covariances with respect to female fitness and the array covariances with respect to male fitnesses. And it goes like this negative relationship. It's even clearer if you do it just based on the ranks of those rather than their actual values. And you see this striking negative relationship. In other words, lines that appear to have the signature of a bunch of dominant alleles when they're for male fitness tend to have the signature of a bunch of recessive alleles when they're expressed in females. which is certainly consistent with this idea of dominance reversals being a big part of this. There's a bunch of caveats here. The most obvious of which being, this is data that comes from just 16 lines. I should say there's a massive amount of work to do these experiments. So it's easy for me to sit back and say, well, they only had 16 lines, but there's a huge amount of work even doing it with this amount. Nonetheless, of course, it would be more convincing and more compelling if there was a much bigger data set. Okay, so let's jump back away from real data for a second and just think about how common should we expect sexually antagonistic alleles or sexually antagonistic mutations to be. So when mutations are occurring in the genome, how often would we expect sexually antagonistic mutations to occur? And then we'll also think a bit about, if we knew how often they occur, we might also want to think about how much do we expect them to contribute to standing variation for fitness, and might it be different in some contexts than other contexts? So Guy already introduced you to Fisher's geometric model. So again, remember the idea here is we imagine 2 dimensions in phenotypic space in this cartoon, but it could be n dimensions. And then the idea is that a population, or you might think of a population as being some point in this plane, and then if you draw, it has this distance from the optimum, and so then a mutation is represented by some deviation from that initial phenotype, and any arrow that takes you inside this circle, because the circle, along this circle are all equally distant points from the optimum. So any arrow that takes you inside the circle brings you closer to the optimum and is a beneficial mutation. Guy already talked about that. So I want to just think about this a bit conceptually when we think about having 2 different sexes. So this is from a model by Tim Connallon and Andy Clark. And now you can sort of think of exactly the same idea, but you have to think about it for both sexes. So here's a female optimum and a male optimum, and where the current female phenotype is and where the current male phenotype is. The black arrow represents the direction of selection, right? And so the black arrow is representing selection, and so if you were to take those 2 arrows and put them on top of each other, you can think about the difference in the angle, the angle represents the difference in the direction of selection being experienced by males versus females. And we can think about mutations that occur. So you can imagine, so that what this cartoon is meant, this red arrow is meant to represent the same mutation in males and females. And this is, they've generalized it here. So here they're imagining that one mutation might have a slightly different effect in males than it does in females, right? So we could imagine that a mutation that affects the phenotype identically when it's in a male as when it's in a female, its phenotypic effect, in which case these 2 arrows would lie right on top of each other and the angle between them would be 0. But in reality, we think I think the existing evidence suggests that mutations affect phenotypes similarly in males and females, but often not in exactly the same way. Okay, so we'll probably have some angle here that might not be 90 degrees or certainly not 180 degrees, but it's probably unlikely to be exactly 0 either. Well, I'm not going to talk too much about this model. These guys did a full analysis of it, but you can think about, you can work through a model like this and figure out what fraction of mutations are going to be sexually antagonistic for fitness, right? And I'll leave you to look at this yourself if you're interested. I just wanted to use this model to think qualitatively about this issue. Sorry, the blue arrows. The blue arrow represents just a different mutation. So the red arrow is one mutation that works like this. A blue mutation affects females this way and males that way. So what one nice kind of simple result is the correlation in fitness effects between males and females will be the product of the correlation in the direction of selection between the sexes, which depends on this angle, and the correlation in mutational effects, which is basically the average of all these thetas across all mutations that could happen. Okay. Which if you think about kind of makes intuitive sense. Okay. So here's one of their results. This represents the fraction of mutations that will be sexually antagonistic. So of those mutations that are beneficial in at least one sex, what fraction of them will be sexually antagonistic? And they plot this as a function of that correlation, and you can see it can be quite high or quite low. And again, it's helpful to kind of draw this out, and you can see why that is. And as a consequence of a large fraction of mutations being sexually antagonistic, there's this cost to the rate of adaptation that essentially the rate at which one sex adapts is much slower than it would be if it wasn't constrained by so many potentially beneficial mutations for that sex being deleterious in the other sex. Okay, so here's a bit of a cartoon where I've imagined here's the fitness optimum for males and the fitness optimum for females, and the circles just represent different contours of fitness as you walk away from those fitness optima. And just for simplicity, I'm imagining the males and females have the same phenotype. So there's no sexual dimorphism in the phenotype. And also for simplicity here, I'm going to imagine that the mutations have equal effect. So, let's think about this. So if we have a random mutation occurring that affects the phenotype, as shown by this green arrow, that would be a mutation that is deleterious for both sexes, right? Takes them both further away from their optimum. This one would also be deleterious for both sexes, and this one would actually be beneficial for both sexes. So actually, all three of the mutations I showed you were what we call sexually concordant. Selection worked in the same direction on them for both sexes. But now let's keep this fitness landscape exactly the same as it is, but imagine this population has adapted somewhat, and it's moved over here. Now let's look at those same three mutations. Well, this one is still deleterious for both sexes, but this one is now beneficial for females, but deleterious for males. And this one is now beneficial for males, but deleterious for females. So if I like going through an example like this because hopefully you get the idea that when both are far from the optima, then you would generally expect to find that a large fraction of mutations are going to be sexually concordant. But perhaps as you move closer and closer to that optima, you're going to get an increasingly large fraction of the mutations being sexually antagonistic. So we were inspired by this idea conceptually, actually several years before that formal model was, and we were thinking about this in the context of the nature of segregating variation, right? So if you have within any population, there will be some segregating variation. And if you imagine the environment suddenly changes, right? So if we imagine we're back here, and maybe the fitness optima, we're close to that point, and then the environment has changed and moved over there, that at that point in time, the existing variation that's in the population might largely be sexually concordant. But once a population has moved closer to the optima, the variation that will be sort of, essentially you use a lot of the sexually concordant variation that was in a population to get to that point. And then the variation that's left might be primarily sexually antagonistic, or at least a larger fraction of it would be sexually antagonistic. Yeah?
So these X's are the traits? So these.
Yeah, that's right. So this is meant to represent some arbitrary trait 1 and arbitrary trait 2.
Do they actually represent the mutation at the more microscopic DNA level?
They represent, so if, let's just to make a silly example, Z1 might represent arm length and Z2 leg length. So we're in an arm length, leg length plane. And then the arrows represent how a mutation would affect arm length and leg length. Yeah, that's right. So that's the way I was imagining it. Those exact same mutations affect the phenotype in the same way, which, so it doesn't have to be that way, but that's the way I'm imagining it here. Yeah, but it's not really a necessary assumption. I guess the idea is, regardless of where you are, anywhere in this plane, there's a spectrum of mutations that are going to send you in any direction. And so, when we were down here, there was those 3 possible mutations that could send you in those directions. And when you're up there, there's some mutations. Maybe they're the exact same ones at the DNA level. Maybe there are different ones because there's, epistasis of how mutations how they affect you when you're currently in this part of phenotype space versus that part of phenotype space. The only thing that really matters is that you have roughly the same spectrum of phenotypic mutations in either of those places. Yeah. I completely agree. The idea is the fraction of mutations that are sexually antagonistic will increase as you move to that point. If you increase dimensionality, as you say, that the fraction might be higher when you're there compared to when you're there, but it can still be very, very small in high dimensionality.
Because there's one dimension where it's antagonistic and the other where it's concordant.
Yeah, I agree. So that's also worth pointing out that the difference between the mutational input and the standing variation, right? So back here, when the population was back here, there was very little sexually antagonistic mutation, and so there probably couldn't be much in the standing population because it wasn't really possible for there to be variance. When you move up onto that line, it's true that in high dimensionality, you would still have a small fraction of the mutations might be sexually antagonistic, but you may be more enriched at the standing variation for those in terms of their contribution to standing variance. So we did a very artificial experiment to test this idea. And this is what we did. We created 2 kinds of populations. One that we called peak populations, so ones that we thought were reasonably close to those peaks. We don't know how close, but we know we're going to say they are closer than the other ones. The other ones we call off-peak, right? And the main idea here is that the peak ones, not that they're sitting right on the peak, but they're closer to the peak than the ones that we call off-peak. So the peak populations were 3 replicate populations, from a larger lab population that had been living in this particular environment. The sort of very artificial way we created these off-peak populations was we took this original large lab population, this is in fruit flies, this sort of large lab population that had adapted to this environment, and we crossed it to another different lab population that I had that was adapted to a different environment. So we made this cross, right, which is essentially pulling that the phenotypes away from the optima, and we created an F2 from that population. So it's a very artificial way of moving this population. But we wanted to do it and see what happens. Okay, then what we did, so we had three of these peak populations, replicate peak populations, and 3 of these off-peak populations. And for each of those, this was work of a former postdoc, Tristan Long, is he took about 150 males from each of these populations, and for each of those males, he put it in a mate competition with some competitor male competing to mate with the females. So for each of these now 6 different populations, 150 males each, he does these 2 competitions, and then he picks out those males that won both of their contests. So we think of those as the winner males or the successful males. And he picks out all the males that lost both of their contests, and those are the loser males, or the unsuccessful males. Okay, so we've got 2 kinds of males, winner males and loser males. And then he takes all of these winners and loser males and mates them to females and looks at the resulting offspring. And this is what he found. So this is the fitness of the sons produced from those crosses. So for the off-peak populations, what he found was that successful males produced much more fit sons than unsuccessful males. And these are 95% confidence intervals. And that's highly repeatable in all the off-peak populations. In the peak populations, we don't see this. Okay? So when you're far from the peak, if you have good dads produce good sons. And bad dads produce bad sons. What about for daughters? Well, for daughters, for the off-peak populations, we see the same thing. Good dads produce good sons, or good daughters, and bad dads produce bad daughters. But for the peak populations, we see the opposite result. In other words, things seem to be sexually concordant for the off-peak populations. Right? For both good successful males produce both good sons and good daughters for off-peak populations. But when you're at the peak, good successful males produce poor daughters, but that seemed to have no effect on the sons. Again, I think I have some figures out of order. This is consistent with this idea of there being sexually antagonistic variation in these peak populations, but that sexually antagonistic variation residing on the X chromosome, right? Because then the fathers, the successful males, will transmit their X chromosomes to their daughters, and if you have sexually antagonistic variants, then the things that have made you a good male will unfortunately make your daughters unfit. But it will have no effect on your sons because you don't actually transmit your X chromosome to your sons. Yeah, that's right. I have to be honest, I don't remember the details of that. I did notice that actually just as I was putting the slides together. I was like, why aren't those ones higher than the others? And to be honest, I can't remember the detail of why that is, because certainly one would expect that. Yeah. So we would expect they would have more sort of total variation, or that would be my expectation. I mean, certainly, in some sense, you can see that, and that these differences are quite large. So that's in some some reflection of that variance. Yeah. I should say, while the thing about the intuition that these bars shouldn't generally be much higher than those bars, this difference in environment is actually in the larval environment, and these are adult fitness assays. So one might still expect it, I think I would, in the sense of they develop in this different environment, and so you might expect, well, if you develop an environment you're not adapted to, you might not be so fecund at the end of that. But it may be the case that the big effect is in terms of survivorship, and there's a smaller effect that comes out in that way. Or there has been some kind of rescaling that I don't remember is the alternative explanation. Okay. All right, so I want to talk about sexual dimorphism. So arguably, the central goal of evolutionary biology is understanding sources of phenotypic variation and how they arise. And clearly, one major source of variation within many animal populations is the variation that occurs between males and females. So just to be clear, sexual dimorphism refers to a difference in the distribution between males and females, and usually we use it to mean the difference in the means between males and females. I always like to give this disclaimer that sexual dimorphism can actually, at least in principle, can evolve for multiple reasons. The most obvious one is that the one that I've been talking about, that selection is pulling males in one direction and females in a different direction, resulting in the evolution of dimorphism. Though in principle, it can happen in other ways. One could imagine that selection is pushing both sexes in the same direction, but for some reason, selection is stronger, say, in one sex than the other. And if mutations don't affect both sexes all equally, say mutation A has a larger effect on males than it does on females, but mutation B has a larger effect on females than on males, whichever sex experiences the stronger selection, well, that mutation is more likely to fix than the other one. And because of that kind of thing, in principle, you could get this difference you could get this evolution of dimorphism, at least in a transiently, until they had both reached, if they were both going to the same optimum and reached that. Alternatively, you could get, even if selection was the same, of equal strength, but you, for some reason, had a difference in the amount of sex-specific genetic variance. So in that case, selection is, again, or in this case, pushing both sexes in the same direction and now with the same strength, but for whatever reason, mutations affecting, say, the male phenotype occur more frequently than mutations that affect the female type. And so then one sex could potentially get ahead of the other, and you would have this dimorphism, again, at least transiently. That being said, well, first of all, it's worth noting that any of those mechanisms require the existence of alleles that have different phenotypic effects in males and females. And I think it's fair to say that in some sense, we're still at the very early stage of really understanding the extent to which mutations affect male phenotypes and female phenotypes differently. Certainly, we have evidence that does happen, and we have some examples, but really quantifying that well, I don't think we've done that much yet. Okay, so while I gave those caveats for those three, or I gave those multiple possibilities, I think most people think that the reason that we have sexual dimorphism is because of a history of past divergent selection between the sexes, pushing males in one direction and females in another direction. But an open question is the extent to which, when we observe dimorphism today, and we observe it in all kinds of species and for many kinds of traits, whether that current level of dimorphism represents evolution having solved this divergent selection, or whether it's only what we call a partial resolution to the conflict. So, here's the scenario we're imagining. Imagine we start with one population where males and females are the same, but the female optima is over here and the male optima is over here. One with enough evolutionary time might expect the two sexes to be able to diverge, right? If you find the right kind of mutations that allow them to move towards their optimum. And eventually, ideally, one might hope that they can each reside under their optima. But in reality, when we look at populations, if we just look at a species, we don't get to see those fitness landscapes, right? Those aren't just readily apparent to us from observation. We can just observe the distributions. And so when we observe a distribution, we don't know if it represents this, a partial resolution where there's still selection trying to pull it further apart, or whether it represents this state of resolved conflict. Okay. So I want to talk about 3 studies that are sort of related to this idea. We'll see if it's 3, depending on how much time I have. 3 fly studies on this. So the first study is one I really like. It's by N.G. Prasad, that some of you might know from IISc. I can't remember which campus is it. It's well, yeah. So this was work he did when he was in Canada in Adam Chippendale's lab, and they had this really nice idea. It was this: that if balanced polymorphisms maintained by intralocus sexual conflict exist, then those allele frequencies would be steady at these intermediate allele frequencies under normal circumstances, right? Because selection in males pushing the allele frequency in one direction, and selection in females pushing it in the others, and so we should have these things sitting around at some intermediate frequency. But if you could magically stop selection on one sex, then one allele would be able to spread quite quickly. So that was their experiment. They created what they called these male-limited selection lines, where only the genomes carried by males were transmitted from one generation to another. In other words, only selection in males mattered to which alleles would go into the next generation. So I'm going to sort of skip some of the more technical fly details of how they accomplished this, but I want to give you the basic idea. So this is meant to represent sort of the entire genome of the fly that starts off being genetically variable. So think of these as haplotypes. So here's haplotype number 1, haplotype 2, 3, 4, and 5. And this M, that haplotype is paired with this other M haplotype, which is kind of it's a sort of a nuisance thing. What we're interested in are these different P haplotypes, which are, I'm using P to represent, to mean paternal haplotype. So the way this works was they started with this genetically variable population, and each generation they would cross it with this stock that we can think of as being MM, carrying these maternal haplotypes. So when you do that cross, you get a new set of haplotypes, but it will be enriched for whichever of these haplotypes carried alleles that were beneficial for males. So when you do this cross, you create all these different kinds of offspring, and what they would do is remove the females. And and so they would remove the females, and they would only take individuals that carried one of these, any one of these parental haplotypes. Okay, so you would, in principle, from this cross also be creating individuals, but they had a way of excluding those. Okay, so the thing that's getting transmitted from the fathers each generation are these P haplotypes, not the M haplotypes are just getting removed each generation. So what's evolving is the set of P haplotypes. Okay? They also did some additional tricks that I'm not going into to allow for recombination amongst these different P haplotypes. Okay, so then the next generation, they would again take the sons out of this cross and again cross them to females from the stock. And with each generation, the selection that's occurring in males is enriching this pool of P haplotypes for alleles beneficial to males. So they do this for a number of generations, and they had four of these male-limited selected populations and 4 of these control populations that they ran for 25 generations, and each consisted of about 1,000 males and 750 females. And I think this is a really striking result. After only 25 generations, so they ran this for 25 generations, and then they can take those flies at the end, and they can take those that's whatever the evolved set of P haplotypes are, and from those P haplotypes you can make more males, or you can make females, right? Remember, every generation you're actually making females, but normally in the maintenance you discard them. Well, at the end of the 25 generations, they said, well, let's make those and measure their fitness. So they wanted to measure the fitness of males carrying those P haplotypes and how those P haplotypes affected female fitness. And that's what's shown here. So in this orangey color is the fitness of males made with those male-limited selected haplotypes compared to haplotypes made from the control populations. And you can see you get better males. So that's a 15% increase in fitness, which is quite a large increase in fitness. And you get the opposite for females. So the haplotypes that when you selected to make better males, the consequence was making worse females. So again, you wouldn't expect this if selection was concordant, but you would expect this if there's a bunch of sexually antagonistic variation that's contributing to this response. Yeah. I don't think you should think of it as going in the other direction. These lines are just to connect the points from the same treatment, right? I mean, the way I think about this is kind of the starting value. I can't remember exactly how they're measuring fitness here. I think they're measuring it relative to a competitor. And so the competitor, how the control does relative to the male version of the control does better relative to the control might be different from how the female does relative to the competitor used for the females. In other words, I think this scale is somewhat arbitrary.
It's only the relative difference.
It's only the relative difference that matters. Yeah, I mean, I think what you're interested in is this value relative to this and this value relative to that.
So, second question is, did they measure these fitnesses for intermediate generations just to see how the fitness changes?
No. Yeah, that would be great. Very difficult to do this in flies at all. And it would be more feasible if you could freeze flies and then, ideally what you want to do is freeze them along the way and then pull them out and measure them all at the same time. This is what Christian and I dream of happening, but we're envious of the worm people and the yeast people for that. So the short answer is no, they didn't do that. So my interpretation of this is this kind of result implies there's quite a bit of sexually antagonistic variation segregating within at least this lab fly population. Perhaps even equally interesting was they looked at some sexually dimorphic traits, one of which is development time. So in flies, females develop a bit faster than males. And so, so here's a case where we have dimorphism in a trait, the trait being development time. And so the question is does the observed dimorphism in development time, does that represent the resolution of past conflict or only the partial resolution of conflict? That there's still selection pulling them further apart with respect to development time, but neither sex can go in the direction it's being pulled because the effects of mutations are genetically linked across the sexes, right? The pleiotropy across the sexes. Well, now they have an opportunity to test that, right? Because if under the hypothesis where there's ongoing conflict, then if I remove the selection from one sex, then this sex is free to go. And if it had been constrained because of the conflicting selection, then it's because the effects are linked across the sexes. And so when this one moves, it should drag this one too. Well, let's look what happened. So here is where females were and males, so females develop faster, so they have a lower score, a lower time to develop than males. And then if you look in the male limited line, so where you remove selection from females, the males move in the male direction over here, and they drag the females with them. And here's another trait, dry weight. Males tend to be smaller than females. And again, the males have moved in the male direction. They were smaller than females to begin with, and they evolved to be even smaller. And when they do, they drag the effect on females with them. And then this is growth rate, which I think is just one divided by the other, if I remember correctly. Yeah. Yeah, I mean, so this is just a weird thing of flies that no one's ever been able to satisfactorily explain to me, that somehow males take longer to develop, and yet they're smaller when they close as adults. Do you have any explanation for this? I mean, there's a story I've heard, but I don't think there's any evidence for it, so I won't bother repeating it. Okay. In the last 15 years or so, there's been an explosion of research on gene expression, as many of you know, and people have been very interested in the fact that you can find many genes, often thousands of genes, that are expressed differently between the sexes. My view of this is sexual dimorphism, just like we see sexual dimorphisms for various other traits. Now we can see it down at the expression level, that it's not sort of a fundamentally different thing than what people have been studying in the past, but because it's an omic thing, people got quite excited about it. So this sexual dimorphism in gene expression is also known as sex-biased gene expression. And so people like to classify genes as being unbiased, meaning expression levels are similar between the sexes, or male-biased, meaning that a gene is expressed at a higher level in males than females, or female-biased, meaning the reverse. And this is where one of the things about gene expression is very nice, because you can view the expression at all the genes across the genome as many thousand phenotypes, as many thousand traits. And so in principle, you can do this kind of thing at the expression level. And in fact, these guys who did this, or at least I think the flies from this, are being used to, I think there's a paper on bioRxiv that's doing this now kind of at the expression level. I think they tend to see this, but I don't, as I told you, I read things and then rapidly forget them, so I don't want to sort of say what they found because I don't really remember.
So in the unbiased, it doesn't move?
I think so. I'm gonna show you some results from another one. I actually think this is a better experiment for that than the one I'm going to show you, but you'll get what you get and you won't get upset. Okay, so I think as a field we're interested in which genes differ in expression between the sexes, how many genes differ, and how strongly they differ. And certainly one thing we know is that all of this sexual dimorphism expression certainly can depend on things like what developmental stage you look at, what tissue you examine, the rearing environment, and which species you're doing this in. So bearing all that in mind, there's lots of reasons that, in other words, there's lots of reasons that sexual dimorphism and expression can vary. And we'd like to understand the reasons that it varies. And certainly we'd like to, one of the major reasons we'd like to understand is, are there evolutionary reasons why the amount of gene expression varies? And that leads me to this paper by Hollis et al. From a few years ago. And so the motivation for this is certainly one reason that species could vary in the amount of sex-biased gene expression is because of the variation in the strength of sexual selection. So the idea being that sexual selection on males is one of the major things that causes selection to favor a different phenotype than in females. And so then if sexual selection was in some way removed or diminished, then males and females wouldn't be selected to have such different phenotypes. That was the kind of the basic idea of this. So in other words, without sexual selection, males wouldn't be selected to be so different from females. And so you might expect males to evolve. You might expect, and I'll return to this point later, males to evolve to be more female-like, predicting a feminization of the transcriptome. So a reduction in the extent to which male-biased genes are male-biased, and an increase in expression of female-biased genes in both sexes. So what they did in this experiment was to create a set of populations, a control set that they've maintained under normal conditions, and then a set where they attempt to remove sexual selection by doing enforced monogamy. So in other words, each generation, rather than letting flies mate and interact and allowing sexual selection occur, they pick a single male and they pair off males and females so that males cannot vary in the number of mates they get. They get one female, so you massively reduce, if not completely eliminate, the opportunity for sexual selection to occur. It's a lot of work to maintain a fly population like this. They had 4 of these monogamy populations, 4 of these control populations, and these are quite small populations, 200 flies per population. Again, the small size is largely because of the amount of work labor it takes to maintain these monogamy populations. Well, here's what they found. So this is the change in expression from whole bodies of flies after 65 generations of monogamy. So they're looking at the change in expression in monogamy compared to the expression in control. And in blue are the genes that that were previously identified as being male biased, and you see that they have that their change in expression tends to be negative. The distribution has shifted in this way. And female biased genes have shifted to the right. So this is this signature of feminization of the transcriptome, an increase in expression of female biased genes and a decreased expression in male biased genes. And that you get more or less that result regardless of whether you're measuring expression in females or measuring that expression in males. That was the data from whole bodies. They repeated it using expression from heads, because it's you can get these changes in expression because of the changes in the relative size of organs, and so to try and reduce that problem, they looked at in particular, people worry about the relative size of the gonads relative to the rest of the body. So here they use the data just from heads, and certainly, at least when you look at the expression in females, you see this effect even more dramatically in females, and there's at least a hint of it when you look at expression in males. That being said, so that sort of like beautifully fits this little, prediction and story, but there are some caveats. So enforced monogamy does more than just remove sexual selection. For reasons that I don't have time to go into, we now know that it actually does some other things. It changes selection on females quite a bit, and it still causes selection on males, but just for different kinds of phenotypes than the selection they experience under control conditions. And I'm happy to talk about that more later if anyone's interested. But it's not quite as simple as I first laid it out to be. So given that, how robust is this prediction of feminization of the transcriptome? That's the result they saw, but I'm not sure that you would always expect to find that. And in that context, this other group performed almost the identical experiment in a different species of Drosophila pseudoobscura, which to most normal humans looks exactly the same as Drosophila melanogaster. So you might expect to find exactly the same results, but their results were in fact very different. So after 150 generations of monogamy, they found male bias genes increased in expression, so exactly the opposite, and that female bias genes decreased in expression. Their results, exactly what their results showed, depended on what tissue they were looking at expression from, but certainly their results were not, didn't line up very well with the results of this previous study. The second caveat I want to point out is in that experiment, and more generally when people have seen in the even earlier studies where people were just documenting dimorphism in expressions, as I said, sometimes you will find thousands of genes, sometimes 40% of the genome would be dimorphically expressed between the sexes. And sometimes that is either implicitly or explicitly interpreted as indicating that there's sexually antagonistic selection across the entire, across all of those genes. And if that certainly may not be the case, right? It could, in principle, it could be a single gene has changed, and that one gene is selected to be expressed differently in one sex from the other sex, and that one gene alone causes a cascade of effects on a whole bunch of other genes. So I think we have to be really careful at looking at dimorphism in expression and interpreting what that means about how prevalent sexually antagonistic selection is across the genome. Because we certainly at this point have many, many examples of dimorphism in expression, but I think we're very far from being able to link that to how commonly there's sexually antagonistic selection on the actual genes, individual genes that are being dimorphically expressed. So to that end, about the true targets of selection, I want to end with one last study that I'm very intrigued by. So this is this paper that came out earlier, about a year ago or during the last year, that is interested in the genetic architecture of loci that might be under sexually antagonistic selection, whether there's many or few loci, whether they tend to occur in particular types of genes, whether they tend to be regulatory versus structural variants, and this kind of thing. So what this group did was they took about 200 haplotypes, sort of full genome haplotypes from a fly population. So by a haplotype, I mean, flies have 3 major chromosomes, so you can think of it as taking at random, a copy of the X chromosome, a copy of chromosome 2, and a copy of chromosome 3 from a population that represents one haplotype. You repeat that process, you have a second haplotype, keep repeating it until you have about 200 of those. And using some tricks of flies, you can then take those haplotypes and you can make as many copies of that haplotype as you like. And so they did that, so they would take haplotype 1 and made it to a bunch of individuals from their source population. So they can look at the average effect of this haplotype on fitness of when it's in a male and fitness of when it's in a female. So they can characterize each of these 200 haplotypes with respect to male fitness and female fitness. So what they're going to do here is a GWAS. And I want to point out, this is a GWAS with 200 haplotypes, which on one hand sounds really good. On the other hand, guy is working with data sets. So how many is in a typical human data set?
What?
How many individuals do you have for a typical GWAS data set?
I don't know typical GWAS, but the golden standard now is UK Biobank, but that's 500,000.
500,000. So we're comparing 200 to 500,000, okay? So bear that in mind. We're going to have a lot less power here. Anyways, they do this. They measure how these 200 haplotypes affect male fitness and female fitness. So that these are all these points plotted here. And then they can define this they do sort of a change of axes. So if you, instead of using the original axes, you do the axes along the diagonal, this axis represents an axis of antagonism, right? If you're over here, that means you have high female fit, you're a haplotype that creates high female fitness, but very low male fitness. And the opposite corner represents the opposite thing. A haplotype here would be high male fitness and low female fitness. So they can, So they can characterize each haplotype with respect to its position on this axis of sexual antagonism, right? So this would be, this would represent the value of this genotype along this sexual antagonism axis, having some negative value, meaning it tends to produce high fitness males and low fitness females. And these two genotypes would have positive values along this axis. So what they want to do is a GWAS, a genome-wide association study on this trait, this new trait they've defined, which is value of on this sexual antagonism axis. And in this way, they hope to find genetic variants that are sexually antagonistic. That's the idea. I had a few slides on what a GWAS was, but I think Guy covered that. So here is the figure that they found. If I remember correctly, when you correct for all the multiple tests you're doing, none of these SNPs are individual SNPs are convincingly individually associated with sexual antagonism. And this is certainly in part because of the much lower power that you would have in a study like this compared to when you're using a big human data set. Instead, they sort of draw this line across which, if I remember correctly, represents a false discovery rate of something like 30%. So in other words, the idea, in principle at least, is 30% of the points above this line are false positives. But still, their hope is that this set of SNPs, compared to the whole genome's worth of SNPs, is enriched for things that are sexually antagonistic. Many of them may be false, but that set is enriched compared to the genome at large. That's the idea. But certainly, you shouldn't believe in any one of those SNPs as having a very high chance of truly affecting the trait. Okay. Right, so as I said, there's no individual SNPs that have strong statistical support. They are hoping that this will be a set of SNPs, the ones that fall above this line, will be strongly enriched for being having an effect. Okay, again, a couple points about association versus causation. So some sites are going to be associated with the phenotype because they have a causal effect on the phenotype, and some sites are going to be associated with the phenotype even if they have no causal effect on the phenotype, but they're correlated with statistically correlated with a site that does. In other words, if they're in linkage disequilibrium with a site that does. And there's much larger linkage disequilibrium concerns when you're using this kind of small lab population than when you're using a much larger data set. Okay. I think we know what linkage disequilibrium is, so I'll skip that. Right, okay, so I'll just skip to some findings. So what were the properties of the SNPs in their list? So even though that list might be, we have to look at that list with a high degree of skepticism, right? But their hope is that it is enriched for things that truly have this kind of effect. Well, one of the things they looked for is whether you disproportionately find these sexually antagonistic SNPs on the X. Remember, we talked about the X being potentially a hotspot for this kind of variation, and the answer is they're not disproportionately found on the X. But again, remember, you have to be really extra wary of this kind of result. This is a negative result, and we're already concerned. We don't know how much of their list really contains true variance and how much of it's kind of just garbage. So maybe the reason you're not finding them disproportionately on the X is because their list actually has a lot of garbage. So this is not really strong evidence against them being found on the X, but they thought they would look and they would try nonetheless, but they couldn't find any. They did find that non-synonymous SNPs are overrepresented amongst the sexually antagonistic SNPs compared to SNPs at large across the genome. So that's kind of interesting. And perhaps equally or more interesting was, even though they found that enrichment for non-synonymous SNPs, they found no support for an enrichment in the regulatory regions. And that was maybe counter to intuition, because I think many people thought or think that when there's sexual antagonism between the sexes, it will more likely be just in expression, that the same actual protein is good to have in males and females, but just maybe the amount of it. Not that you would actually want a different protein sequence in one sex than another. And bearing all the caveats I said earlier in mind, that's not what they see. I can't, to be honest, so the 2400 represents all the SNPs above the line. At some point, they just take one thing per peak to come up with a more reasonable number. And I can't remember when in this set of results that I'm showing, they're using the full set of all 2,400 versus the, one site per peak. Right, right. Well, so they also look for an enrichment in synonymous. You don't find an enrichment in synonymous. Yeah.
Okay.
I'm 98% sure they also had synonymous ones that they looked at. The next thing was they looked for whether their sexually antagonistic SNPs tend to fall in sex-biased genes. Right, so you might think that if these genes that are differentially expressed between the sexes are under antagonistic selection, that your sexually antagonistic alleles would fall in those kinds of genes. But they don't. In fact, they found that that unbiased genes were more likely to harbor sexually antagonistic SNPs than either male biased or female biased genes. If you were to, again, please take all of these results with serious caution. I think the results are really intriguing, but they also should be viewed with a high degree of caution. But if you take this result at face value, one interpretation of that would be, oh, well, the sex-biased genes represent resolved conflict, and it's these unbiased genes where selection would like to have differential expression in the two sexes, but hasn't been able to achieve that, and so that's where you find these sexually antagonistic variants. That would be an interpretation of this result, if you were just to take it at face value. They also for some reason, people have sometimes predicted sexually antagonistic variation would be found more commonly in genes that are more pleiotropic or genes that are expressed in a broader range of tissues, and they didn't find that. Again, especially with the negative results, you should be suspect because we don't know we don't really know what fraction of the sites within their list are actually true positives. The part that I find most intriguing of all, it's hard for me to imagine how they can pull out accurate SNPs with 200 haplotypes, especially when we know that this is going to be really hard to do. The next result is one that makes me think maybe their set of SNPs really are enriched for true things, because I at least can't think of another explanation for it. So this is the part I find most interesting. So they looked at their sexually antagonistic SNPs, and essentially their idea was, hey, if these things are under balancing selection, because of sexual antagonism, they're under balancing selection, then that polymorphism should be polymorphic in natural populations. And better than that, well, so they and they compared that to as a control for that, they compared that to non-sexually antagonistic SNPs in their sample that were at the same allele frequency. Because you want to control for allele frequency. They also control for a couple other things as well. And yet when they looked in these natural fly populations, they found that their SNPs were more likely to be polymorphic than this control set of non-sexually antagonistic SNPs that were matched for similar allele frequency and some other features. And perhaps even more compelling than that was they said, okay, forget about looking within Drosophila melanogaster, let's go look at Drosophila simulans. Do we find these same polymorphisms in Drosophila simulans, which might indicate that these polymorphisms are actually really old, that they're shared polymorphisms from 1 million years ago. And again, they found that their sexually antagonistic SNPs were indeed those exact same polymorphisms were more likely to be found in Drosophila simulans than was the case for a matched set of control SNPs. And I really think that's an interesting and intriguing result. Did you?
Tell me if I'll stop asking questions if you're lacking time.
I think this is my last slide.
Okay, so how many SNPs did they have above their significant threshold?
They had 2,400, and then they somehow pared it down to, I think, 250 or something for this part of the analysis.
So 250, and polymorphism levels in simulants, I think, are like, what, like 1%? If you're,
Yes, I think something like that.
So I guess what I'm asking is like, how much of a difference in numbers are we talking about here?
Right. I don't remember off the top of my head.
I see. And this original, I don't remember. This was done in Milan, I guess?
Yeah.
Well, so there's a lot of, kind of like shared polymorphism.
Yeah.
In Milan, I guess.
Yeah.
But, yeah.
So of course, it isn't just that they're shared polymorphisms, they're more likely to be shared polymorphisms for these SNPs than they are for those.
I get that. I'm just trying to, kind of like think about, what numbers are we talking about?
Yeah.
If it's like, a difference between, carrying 7 and carrying 4.
Right. That's a fair point.
Even though it's statistically significant, you can imagine how, just coffee.
Yeah.
Keyboard could do that.
Yeah. Fair point. Fair point. I don't know the answer to that. That's worth looking into. So they find this when they do this comparison to Drosophila simulans, which is 1 year separated, but not when they don't find that difference when they compare it to Drosophila jacoba. And so they argue that these results are consistent with these variants being maintained by balancing selection. So that's a bit of a little vignette into this area of sexual antagonism. From these kinds of experiments and this first kind of GWAS-y kind of approach to it, there's at least, certainly the experiments seem to indicate that there's evidence of this kind of variation. I'm very interested in this classic question of what maintains genetic variance in fitness and the extent to which sexual antagonism might be contributing to the maintenance of genetic variance and also constraining the adaptation of each of the two sexes. But it's, I think it's an open question to, really how important is this in the maintenance of genetic variation in general. And clearly, Guy has a different view on this than I do, so it's been a real pleasure talking to him about this. And that's it for me. So thank you all.